The human upper digestive tract operates like a highly efficient intake facility. As soon as food enters, our biology races to break it down and absorb every available nutrient. This rapid absorption is an evolutionary trade-off. When early humans mastered fire and began cooking starchy foods, the heat partially pre-digested their meals. That external head start allowed our ancestors to grow massive brains, but in exchange, our digestive tracts shrank. Here is the exact biological tool driving this rapid digestion: an enzyme called salivary alpha amylase, or AMY1. This enzyme specializes in dismantling the coiled helices of cooked starch, breaking chemical bonds to release single glucose molecules immediately. Because of these rapid-acting enzymes, the highly processed starchy diets we eat today are completely broken down and absorbed long before they ever leave the upper digestive tract. By extracting all our energy so early in the digestive process, human biology inadvertently creates a massive resource deficit further down the supply chain. Moving further down into the large intestine, we enter a massive, distinct environment. This lower gut is a dense ecosystem, home to trillions of beneficial microbes waiting for their share of the meal. But because our modern diets are fully absorbed upstairs, this microbial ecosystem is left starving. Without raw structural resources flowing down to the large intestine, we lose crucial biological diversity. To keep these lower gut microbes alive, we have to send them complex, resistant fibers that can successfully evade our upper digestive enzymes. Looking at this comparison, the solution comes from the ocean. On the left is a simple, easily digested land plant cell wall. On the right are marine polysaccharides. Found in brown, red, and green seaweeds, these dense structures are forged in harsh oceans to be incredibly tough. By reintroducing these highly complex marine carbohydrates to our diet, we bypass our own early digestion and deliver intact resources directly to a starving colonic ecosystem. Human digestive enzymes are physically incapable of breaking the unique chemical bonds found in marine superfoods like alginates and fucoidan. As this video shows, these marine polysaccharides pass through the caustic, extreme acid of the stomach completely intact, without dissolving or losing their physical shape. When this intact cargo finally arrives in the large intestine, the microbial community responds. Specific bacteria deploy specialized genetic toolkits, known as polysaccharide utilization loci, or poles. This diagram illustrates how poles function. They act as precise mechanical protein receptors that capture large marine sugar chains, lock onto them, and snap off specific geometric pieces. Once cleaved, the bacteria immediately pull those pieces across their membrane for internal processing. Our human bodies lack this unloading machinery. Instead, our microbiome acts as a specialized deep processing facility dedicated to handling complex foreign cargo. Inside this processing facility, certain bacteria act as keystone microbes. Think of them as the master chefs performing the heavy, difficult work of slicing the largest marine fibers into smaller pieces. This creates a chain reaction of sharing, a biological process called syntrophy. Other microbial species survive by feeding off the soluble leftovers created by those keystone microbes. This cooperative cross-feeding ensures that the entire community stays fed, preventing any single bacterial strain from monopolizing the premium marine resources. If a gut community doesn't have the tools to digest seaweed, it can acquire them through a radical evolutionary process called horizontal gene transfer. In this animation, we see exactly how that happens. Human gut bacteria connect directly with environmental ocean bacteria, passing a glowing ring of genetic code that causes the receiving bacteria to instantly sprout new digestive tools. The intake of marine polysaccharides transforms the gut environment, expanding the community's genetic library and enforcing a high level of cooperation between species. The microbial fermentation of these marine fibers yields highly valuable metabolic products known as short-chain fatty acids, or SCFAs. This flowchart maps the three primary SCFAs. Butyrate points to the colon wall, propionate to the liver, and acetate enters the bloodstream. Butyrate fuels colonocytes, repairing tight junctions to seal the gut wall and prevent inflammatory leaks. This metabolic messaging triggers mucin 2 production. This creates a thick, clear mucosal tablecloth that acts as a smooth, protective barrier over the cellular surface. Through the breakdown of seaweed, the microbiome manufactures the exact biological currency required to rebuild and fortify our intestinal defenses. This marine menu selectively shifts the entire microbial balance in our favor, promoting elite beneficial species like Akkermansia. By feeding these beneficial species, the system actively starves out toxic, inflammatory microbes. This process reverses the chaotic state of biological imbalance, known as dysbiosis. A sealed gut barrier connects directly to systemic health. By blocking inflammatory toxins from leaking out of the gut, the body avoids full-scale oxidative stress. Simultaneously, specific marine compounds trigger the release of protective bile acids. These acids travel out of the gut and actively silence inflammatory pathways in our fat cells and liver. By eating resilient marine biology, we bypass evolutionary limitations. We deliver the precise raw materials our internal ecosystem needs for whole-body metabolic health.